presentation. Can you see my screen?
Yes, we can see your screen and hear
Okay.
you well.
Nice. Thank you. So thank you for the presentation. My name is Karl Baltazart, and I'm a first-year PhD student in bioinformatics at the University of Lille. And I would like to introduce you the first result of my PhD in a topic titled In Silico Methods for Deep Time Protein Sequence Reconstruction. So my work is a part of the recognition of fossil taxonomy using the paleoproteomic methods of ZoomMS, which analyses are done on type 1 collagen. You have two ways to do ZoomMS. You have the liquid chromatography double MS method and single MS method. So the difference between both is the number of ionization you do. But they, however, perform in common the trypsin digestion on the collagen, which permit to cut the protein in peptides, then used as marker peptides for taxonomic identification. So as a reminder, I don't know if it's useful, but as a reminder, a marker peptide is a peptide that is conserved in a sufficiently large number of species, but still show diagnostic variation between species. So you have on this slide an example with the P2 marker database for which you can see different degrees of differentiation between species here. So the mass of a peptide is directly related to its sequence, and when you obtain a mass spectra from a fossil, you can directly make inference from the peak, which gives mass indication, to the sequences in the peptide marker databases. The fossil is then attributed to the taxa, which has the most marker in common with him. And the question is how to identify peptide markers from the fossil. So it's essential to have marker peptides because they permit to make taxonomic inferences. If you remember, there is a difference between the double MS and the single MS-MALDI methods. In addition to spectrum data, the double MS returns sequence information, even if it's more time-consuming and more expensive, which is not the case with the simple MS method. You don't have access to the sequence. So in this work, I developed bioinformatic methods that allow the reconstruction of fossil data from MALDI spectra in the context of ZoomMS. The question is, can we predict by bioinformatics the sequence of fossil marker peptides? And we can cite two major interests in this work. So the first one is the enrichment of marker peptide databases and the better knowledge of the evolutionary context for marker peptides. So I developed a workflow in bioinformatics which permits to answer this question. Initially, we will recover MALDI mass spectra and collagen sequence data from current species. The fossil organism has its own evolutionary history, and making inferences on its taxonomy only with current species can only provide partial results. So to pass through this problem, we reconstruct ancestral sequence from current organism using PAML software. So you can see on this picture that the ancestors are located at the nodes of the phylogenetic tree, and their sequences are inferred by a method well known in biostatistics, like maximum likelihood. So this common ancestor is genetically closer from the fossil than the current sequence is, and this reconstruction is our first step of our workflow. Once we have obtained these ancestral sequences, we carry out an in silico digestion of all the sequence data, so the current and ancestral, to obtain marker peptides. And we digest also modern sequence if needed. This digestion is made by software, Pampa, which is designed in our research team at Lille. So we carry out a first step to assign the marker peptides generated in silico to peaks in the fossil spectra. So we have three types of output data. We have peak associated with sequence data, which correspond to correspondence between spectrum information and peptide marker database. We have also orphan markers, which mean that no peak of the spectrum correspond to a sequence of a peptide marker. And also we have orphan peaks, which are peaks whose mass doesn't correspond to any sequence of peptide marker. So it's on the later data that we will be working on to generate new markers corresponding to the peaks of the fossil spectrum and see if we can associate the orphan peaks and marker sequence data. So here it will be the last step of my workflow that I will develop just after. So we have built a model to create variations in the sequence to explore the space of sequences structurally close to our orphan markers. So we can represent this search as the circle in pink here in the picture, corresponding to the space of the sequence explored. So this variation generates new masses, which we compare a second time with the peak of the fossil spectrum. The variation generated are generated at position already known to be invariable within the collagen, because certain positions are conserved within peptides. As you can see for the P2 marker again, in black you have conserved position, and in colored you have variable position. And we also prioritize certain substitution using a score matrix, because the sites that vary don't present all the amino acid possibilities, as you can see here in the logo sequence. When you have a variation site, you don't have all the possibilities of amino acid. Sorry, I will now present the results of this study. I tested this method on 2 different taxonomic datasets. So the first one is concerned the group of Xenarthra. So Xenarthra is a superorder of mammals, which includes current sloths, armadillos, and anteaters. And here we have 13 fossil species and 3 ancestral sequences. So in this table at the right, you can see green boxes corresponding to accurate prediction of marker by the workflow. The letters A and M correspond to a prediction made by the ancestral or the modern sequences. You can see here that the workflow provides additional information, because in the case of the P2 marker here, the prediction was made using only the ancestral sequence for 2 taxonomic groups, which are the superfamily of Megalocnoidea and Megatheroidea. The second data set concern the taxonomic group of Eulipotifla, which is also called Insectivora. And inside this group, you have a family, which is called Nesanfantidae, which is fossils. And for this dataset, I have 7 fossil species and only 1 ancestral sequences. You can see on the table on the right different results. So we have been able to detect fossil peaks just with ancestral or current sequence or current sequences, but also by generating new sequences from orphan marker. You can see with the letter G here. And sometimes several different sequences have the same mass and correspond to a peak in the spectrum. These are known as false positive, but they are only found in small quantities for the marker generated, for the marker F, G, and P1. You can see the false positive here. There is no false positive for the P1. So to conclude, I would like to mention here that the programming language I use is Python. And in addition, as I partially mentioned during the presentation, my work is based on the Pampa development project from our research team, which enables taxonomic attribution of spectra from sequence data. So if you are interested, you have the website link and the GitHub link on this page. As far as my work is concerned, I'm currently testing the program on new datasets, in particular fossil armadillos, for which I have mass spectra. And I'd be delighted to work with your data if you have any. because the problem is to find fossil spectra. If you have some, I will be happy to work with. And finally, I'd like to thank my thesis supervisors, Hélène Touzé and Céline Pou, who have given me a lot of support until now. Thank you for your attention.